Hello students, myself Popere KS, Assistant Professor in Botany at Ray Shikshan Sanstha's RBNB College, Srirampur. In this lecture, now we are going to start with the other organelle, that is the lysosomes. So, lysosomes it was lyso means the term lysosome means, it is a Greek word. Lyso means digestive and zome means body. So it is a digestive body. It was first identified by or discovered by a scientist discovered by the scientist, that is Christian D. T.Y.B.Sc. Now these lysosomes, they are also called as suicide bags. They consist of hydrolytic enzymes or the enzymes which are acidic in nature. So they consist of different hydrolytic enzymes, and mostly it is approx 36 different hydrolytic enzymes they are present in the lysosomes. And this lysosomes, they are dense granular vesicles. So these are dense granular vesicles. So this is about the introduction of the lysosome. Now we will see the morphology of the lysosomes. So the lysosomes, these are mostly, it will be found, suppose this is a cell. So this lysosome, they will be present, they are diffused into the cytoplasm, or they are present as vesicle in the cytoplasm of a cell. Inside, this is a nucleus. So these are diffused into the cytoplasm of a cell, and they will contain the different enzymes or the hydrolytic enzymes. So this is the structure of the lysosomes. Now they are mostly, they can be variable in shape, that is they may be rod shaped or spherical shape or irregular. So they are spherical, rod shape, or maybe irregular. Now they exhibit a great polymorphism. So they show a great polymorphism. Now we will see the different types of the lysosome. So first is the primary lysosome. These are also called as storage granules. So now this lysosome, they are the newly formed lysosomes that do not participate in any cellular activities. So these are the newly formed, and they will not take part in any cellular activity. So they are called as storage granule, because only storage function will be performed as they are newly formed. Now next to this, second is secondary lysosome. These are also called as digestive vacuole. Now these are the lysosome, the secondary lysosome, in which the phagosome or the pinosome, when it fuses with the primary lysosome, it will form the secondary lysosome. So phagosome or pinosome, these are the vesicles. These phagosome and pinosome, these are the vesicles which are containing the material. When it fuses with the lysosome, that is the primary lysosome, then this secondary lysosomes are formed. And in this, the hydrolysis of the material which is present in this vesicles, this is carried by an enzyme, that are the hydrolytic enzymes. So hydrolytic enzymes, they are present in the secondary lysosome. Now next to that, there is the next lysosome, that is the residual body. Or this is also called as post lysosome. Now here, this residual body, after the degradation or the hydrolysis by this hydrolytic enzyme, the leftover part or the debris or the waste material that will be present in this residual body, they are also called as post lysosome. So in this lysosome, there is presence of debris and the undigested material. Now this debris or the undigested material which is present in the cell, if it remain in the cell or in the lysosome for a long period of time, then it is causing, or it is an important factor which is causing the aging. So aging is caused due to the when it is stored, the residual body, if it is stored in the cell for long period of time, so it will be causing, or it will be a reason for the aging. Now next to that, next is autophagic vacuole. When the cell feeds on its own intercellular organelles such as mitochondria or ER, so that vesicles or that lysosomes that are called as autophagic vacuole. So in this autophagic vacuole, the cell will start, it will feed on the intracellular organelles, that is ER or the mitochondria. So this is about the morphology of the lysosome. Next we will see the ultrastructure of the lysosomes. Now ultrastructure, it is mostly very small structure, as we have already seen. It will be diffused in the cytoplasm. So these are the lysosome body. If we see one particular lysosomes, it may be spherical, rod shape or irregular. So this is the, suppose this is a lysosome, so it will have an outer membrane or only one membrane is present. So it is a single membrane structure, and inside there are present of different presence of different granules, or there are present of different protein or enzyme. So this is the ultrastructure of the lysosome. It will mostly consist of the enzyme, hydrolytic enzymes or the proteins. Now the lysosome, the size of the lysosome is from so the size is 0.2 to 0.8 mu in size. It will be, the shape of it is variable. We have already discussed that it may be spherical, irregular, rod shaped, and it is bound by a single unit membrane. So this is about the outer ultrastructure of the lysosome. Next we will see the chemical composition of the lysosome. So firstly, the lysosome enzymes, they are consisting of the different enzymes that are called as hydrolases. So it has presence of various enzymes, and that are collectively called as hydrolases, and they work in acidic medium. Now there are lysosomes, it has acidic medium, means the pH will be 5. So that is highly acidic. The lysosome, the interstructure or the inside there is a pH that is very high, that is equal to 5. And why this has pH 5? Because on the membranes of the lysosome there are ATP dependent proton pumps which are present on the membrane of the lysosomes, and due to which there will be accumulation of the H+ or the proton in the lysosome. So they have proton pump, due to which it has the acidic medium, that is pH is equal to 5. Now there are different enzymes which are present in the lysosomes. The first is nuclease. It will digest, nuclease will digest the nucleic acid. Next is protease. Next is protease, which will digest the protein. Next to that is phosphatase. Which will digest phosphate containing compounds. Next is glycosidase. next is glycosidase, which will the enzymes which will digest the enzyme that digest the compound, that is the carbohydrate compound. Next to that is sulfatase, which will digest the sulfur containing compound or the enzyme. So these are the different enzymes, that is nuclease, protease, phosphatase, glycosidase and phosphatase. This will be present in the lysosome. Now for this activation, so for the activation of this enzymes, there will be requirement of a specific pH, that is the acidic pH, which will be created in the when these ions are present. So under the critical conditions or the critical levels of these ions, these other enzymes will get activated because they are provided with the acidic medium. Now next to this, we will see the functions of the lysosomes. So the main function of the lysosome is the digestion, or the digestion of the unwanted material or the waste which gets accumulated in the lysosome from the cells. So cells thrown out material is digested in the lysosome. So first function is extracellular digestion. So this extracellular digestion means substance outside the cell which are present outside the cell, that will be digested by the release of the enzyme from the lysosome. So substance which are present outside the cells, that is outside the cell. So the lysosome, it will release some enzymes outside the cell. So all the things which are present outside the cell will be digested. So this is extracellular digestion. Next to this is intracellular digestion. So in this type of digestion, during the starvation conditions, lysosome of the our own cell, it will start digesting the cell organelles, and it is done by two ways, that is so the digestion, intracellular digestion, it is done by two ways, autophagy and heterophagy. In this condition, during the starvation condition, the cell will start digesting the own organelles, the cell's own cells organelles. So that is called as intracellular digestion. Next is the inducer of cell division. So inducer of cell division means when the breakdown of the lysosome in a cell, it will take place, so it will trigger or it will mark the activity or the mitotic division to start. So as soon as the lysosome, it will break off, or breakdown of the lysosome, it will be resulting in the mitotic cell division to start. So this is lysosomes. They are called as the inducer of a cell division. Now it has also role in So it has a role in seed germination, because seed has a outer covering, that is hard covering, and it is consisting of the allulon body, or we can say it has a presence of starch. So allulon layer, it is digested by the lysosome. So lysosome involved in the hydrolysis and the removal of the proteins and the starch which are present in the seed during the germination. So it has a role in the seed germination. Next is lysosome cause certain diseases. So due to lysosome, also due to malfunction or overfunction, it also cause certain diseases. That is, one is Popere disease. So it is caused as the high accumulation of the glycogen it will be occurring in the liver, due to which this Popere disease is called. And in the lysosome diseases, they are mostly due to the overfunctioning or the malfunctioning of the lysosome. So this is about the functions of the lysosome. This is all about the lysosome, that is the introduction, morphology, ultrastructure and the functions of a lysosome. Thank you.